Hello, my name is Teddy Herriman. I'm a first-year grad student in the Chemistry and Biochemistry department at Montana State University. Today I will be discussing the comparative complexity of cells, in particular the complexity of microbes. So that is the difference between prokaryotes, archaea, and bacteria versus eukaryotes, but more specifically in this case protists, the single-celled version of them. Now, I would argue that the goals of measuring complexity or comparing it between cells are at least two-faceted. The first component would be to constrain phylogenies. At the moment, there are 3 to 4 phylogenies that are cited. The difference between these phylogenies are not just their results, but the methodologies that they use. For instance, many use amino acid sequence comparisons. Some compare protein folds, others compare information content, so on and so forth. The issue that they all seem to have is how to appropriately, quote-unquote, root the tree, which is to say where do cells line up in terms of being in relation to a last universal cellular ancestor, and how are they specifically related to each other evolutionarily. On top of this, there's an issue of adding viruses into the domains and figuring out where they fit within the tree of life. The big issue here, of course, is viral genomes are so small that even if you're comparing amino acids or protein folds, there's not a great way to compare them to the much larger genomes of cells. Now, the second one, which is especially important for origins of life, is to constrain identity and properties of the progenotes versus the last universal cellular ancestor, which is to say, can we say something about the simplicity and or complexity of LUCA and identify which biological features are quote-unquote simple and or complex and therefore should be expected as the first form of life. With this in mind, I want to discuss some assumptions of biological complexity that are either implicitly or explicitly stated within the literature. The first and foremost, which is very key to origins of life and astrobiology, is the idea that complexity increases over time, which is to say we assume that the ancestral biosphere is less complex in a measurable way than the present biosphere, and that ancestral organisms are also less complex than present-day organisms, with some numeric value of complexity assigned. The second component is that life is complex. So we assume traditionally that non-life is less complex than life, and that this is a key identifier of what is living versus what is not living. As I alluded to, it's important to have an exact mathematical definition for what you mean when you say complex. In this case, I'm going to be using what I call temporal information density, or TID, which is the amount of Shannon information it takes to describe an object, whether that's a biological object like a cell or other inanimate object, divided by that object's overall mass, times the amount of time you are actually looking at that object. Now, the units for that would be number of bits per gram second. For simplicity's sake, bits, you can think of how much space it would take to describe that object on a hard drive divided by its actual physical size. So implicit in this derivation is the idea that size is just size, and something can be big and simply big and not necessarily complex. So for instance, if I were comparing computers, I would be first curious to know how much information it would take to describe that computer, and then I would want to divide it by the computer's overall size, which is to say the smallest computer that can produces the same functionality with the same amount of information to describe it would be the most complex. Now, to understand the difference between prokaryote and protist complexity, it is important to understand the history and the context of where claims come from. I would argue the first statement of prokaryotes being simpler than protists began with Edward Shatton in 1925 when he first coined the terms prokaryote and eukaryote. In those terms, prokaryote is Greek for essentially before the nucleus, and protist means true nucleus. In that he established one came before the other, and also implicitly and to a certain degree explicitly implied that this was because prokaryotes were simpler, which based on his microscopy made a lot of sense. Since that time, a number of other measurements have been used to claim protists' higher complexity. Among them are larger genomes / more proteins. The issue, of course, is known today that if we were to assume that this is the case, you'll have many logical fallacies, such as human beings would be less complex than a rice plant. Somewhat more recently, information content has been used to claim complexity. In at least 2 examples, with sample sizes under 11, 2 authors have claimed that total information content can be used to measure something's complexity. However, I will mention that even Kunin in his paper along these lines mentioned that prokaryotes appeared admittedly better designed. The third way that people claim complexity that I've at least found is that it's because eukaryotes have organelles, whereas prokaryotes do not. This today we know not to be true. Prokaryotes do in fact have organelles. It seems the large difference between the types of organelles they have is that prokaryotes tend to have protein-based organization, whereas eukaryotes depend heavily on their lipid-based organization. And finally, is the idea that you can associate eukaryotic multicellularity to protist complexity, in the sense that protists could have the potential to evolve into multicellular organisms, whereas prokaryotes can only superficially do so. This last point should be addressed further and can be addressed from a simple thought experiment, which is, could multicellularity evolve in the absence of prokaryotes? The answer is no, at least from an ecosystem and biospheric perspective. Multicellular eukaryotes are 2-component machines. This has been addressed by the idea that if we were to eliminate all of the prokaryotes on Earth today, biogeochemical cycles, nitrogen fixation, and many of the phages that we have that regulate our ecosystems would disappear. In particular, with the loss of nitrogen fixation, you could expect photosynthesis to die out within a year. On top of this, there's an even older idea, which is the energy pyramid, sometimes known as the trophic energy pyramid, which is to say that multicellularity is regulated from the bottom up. If we were to take prokaryotes out of the picture and be left entirely with protists, as we'll address a bit more on the next slide, there would not be enough energy for multicellularity to both evolve and then sustain itself. Now, there are in fact some reasons to assume that the dogma of prokaryotes' simplicity in comparison to protists might actually be incorrect. First and foremost is that protist adaptability is not as good as prokaryotes' ability to adapt. How this can be measured is by the number of niches that protists occupy in comparison to prokaryotes. What do I mean by niche? I mean the number of environments they inhabit and also the metabolisms that they are actually capable of. So for instance, prokaryotes are abundant within deep-sea hydrothermal vents, whereas at least single-celled eukaryotes are unable to live in such environments. In addition, while protists only have access to respiration and photosynthesis as far as we know, prokaryotes have many other forms of metabolisms that allow them to access other environmental niches. As a side comparison to this as well, it is pretty well accepted that prokaryotes are more adaptable in the sense that we can also throw them into, say, the vacuum of space, and they will be able to adapt, at least to a certain extent. The second point is that protist metabolic speed is less than prokaryotic metabolic speed. And this is true for both respiration and photosynthesis by mass. Now, if you are to accept that dissipation of energy, as many do within the astrobiological community, is fundamental to life, then it would seem odd that the species that is slower at dissipating energy would be determined as being more complex. In total, with both of these, we could almost assume that adaptability and dissipation of energy are not actually very much fundamental to biology, and or they're not fundamental biological indicators of evolution. Last but not least, that protist biochemical TID is potentially lower than prokaryotic biochemical TID. TID, of course, being the measurement of complexity I presented earlier. I can gauge this by the lower proportion of lipids and lipid organization within prokaryotes. Lipids, by Ivan Bachner, were entirely left out of his mathematical formulation because they were considered too entropically simple to even include. In conclusion, complexity deserves to not just be an abstraction, but a numerical value that we can use to compare biological entities as well as non-biological entities. And so doing so, we might be able to have a better understanding of how life evolves and potentially what life evolved from. As it stands, there's a dearth of literature in regards to biological complexity, and there's even less literature that actively compares living organisms. At the moment, there also appears to be some indicators of fundamental biological ideas that indicate that it is possible that prokaryotes are in fact just as complex, if not more complex, than protists. I thank you for your time. If you have any information regarding complexity, context I might have missed, and or would like to discuss prokaryotic complexity, my email is below. Thank you for your time and the opportunity.